Canvas. If you go ahead and watch that and make some annotations, there are spots in the video where I ask a question and you can make an annotation and that'll count as your in-class participation. So if you could do that before class starts on Wednesday, that would be awesome. Okay, so the first section, and I know it looks like a lot of sections, but I promise most of them are not very many slides. So I try to break it up into chunks so it's not so overwhelming, like here I have 100 slides I have to look at. Now you can look at smaller groups. So this is, if you got an older edition of the textbook, that's totally fine. This is newer information that is not in the older edition of the textbook. So it's on the human microbiome. And the microbiome is a really cool area of microbiology. It's something that I'm actually interested in, not human microbiome, because yuck, people. I like birds, that's my fun fact. And so I like to do bird microbiome. But the human microbiome, the microbiome of anything, is the community of organisms. Most of the time we're talking about bacteria, but it can include archaea, it can include fungi, it can include protists, and it can include some of the viruses, especially those associated with the bacteria. In humans, what older statistics have said, so if you maybe an older BACT textbook or something like that, and they say that for every 1 cell of you have 10 cells of microorganisms. It's been revised down. Now, we think it's more 1-to-1 or 2-to-1. But at least for every 1 cell of yours, you have 1 microorganism that's hanging out with you. Almost every part of the human body does have a microbiome. There are a few regions that are sterile. That would be your brain, your pleural cavity, your bloodstream should be sterile, your bladder should be sterile. But places like your skin, your GI tract, anything that's exposed to the external environment, and that includes your GI tract because you eat, your upper respiratory tract, you're going to have microbes there. The GI tract tends to be the most diverse, particularly when we're talking about the large intestine. But every individual is going to be different. And so the microbiome signature for each one of us is going to be different. We all have different diets. We all like to eat different things. We use different soaps and shampoos. We have different hygiene habits. We have different pets. We live in different places. We touch different things. So while there are going to be a lot of organisms that are important that we all have in common, our microbiomes are also going to be different from one another. And those organisms that we share are called our core microbiome. And so these are the organisms that we would find in about 95% of individuals. We can't really say that every individual on the whole planet has this one same organism, right? There's always exceptions. So if a species is found in about 95% of individuals, we would call that part of the core microbiome. So what I want you to think about for just a minute is, what are the implications of that? Why is that important that there are organisms that we all share? What do you think that means about the function of that organism? Anybody want to hazard a guess as to why there are some species that are really highly conserved in our microbiome?
They facilitate something within us?
Yeah, they facilitate something probably that we need, right? Perhaps they produce a compound that we need that we can't produce. Or they break down a compound that we ingest that we can't break down. So for example, in cattle, in their stomach, they have archaea and protists and bacteria that break down plants. So cows eat a lot of grass, but cows themselves actually cannot digest the grass. It's the microorganisms that live in the cow's gut that digest the grass. So if we're conserving these organisms among all of us, they have to provide some sort of function that is really important. There are other species that we would consider variable or part of the secondary microbiome. And that's going to be because of our different experiences. We live in different places. We've traveled to different places. You may be a vegetarian. You might eat whatever you want. You might be a vegan. You might use this soap versus that soap. And then the other thing to think about is that we may have individuals that have different microbiomes in terms of taxonomy. So there are different species, but they have what we call functional redundancy. They do the same thing. And that could be important because if you have, 6 species of microorganism that do the same function, as long as you have one of those, you're probably okay. You don't need to have all 6. And so in a situation where maybe a person has taken antibiotics and that can disrupt the microbiome, if they have some members that can still do the necessary function, they might be okay. If you wipe out all of the members that do a necessary function, that might be a bit of a challenge for you. And so what we tend to see, and we'll talk a lot more about this as we get into bacteriology, but what we tend to see is that when individuals take antibiotics, they can end up with some secondary consequences. Diarrhea is pretty common with antibiotics because it wipes out the normal gut microflora. Yeast infections in women are pretty common because they wipe out the normal vaginal microbiome, allowing the fungal organisms to take over. So it's important that we have the functions and the diversity in that microbiome. And if we look at a typical human, people know a lot about the human microbiome. There have been tons and tons of studies about the human microbiome. So scientists can really tell you, we know what organisms are found where. And we often know what those organisms do, what we think they should do. What a lot of scientists are working on now is understanding how these organisms that we carry around with us can affect things like our immune system, our thought processes, right? There's this gut-brain axis. If we have a disbalance, people propose that if we're imbalanced in our gut microbiome, that could lead to things like depression. Other skin conditions. And so now we're trying to really learn how the microbiome overall interacts with us. And we kind of have to think about it that way because we all carry these organisms with us. They affect our phenotype. Right? If you have the wrong microorganisms, you could have on your skin, you could have things like eczema, inflammation. You could have in your gut, you could have diarrhea. You could have respiratory conditions if you have the wrong microbes in your respiratory tract. So it affects your phenotype. Eventually, if we're affecting the phenotype, do we affect, this is kind of the evolutionary theory, do we affect the evolution of the organism? So do our microbes drive evolutionary processes? And so these are all things that we have to think about when we think about the microbiome. So those of you interested in going into clinical medicine, you really have to be cognizant of your patient's microbiome. How can it be affected by whatever therapy or treatment you're doing to your patient? How can you help them recover? Right? All of those things you have to think about. So the normal flora or the normal microbiome is the person's standard condition. Every individual has a unique community. Every body site within that individual has its own unique community. Moist areas on the body are different than dry areas, or different than internal areas, different levels of oxygen. Why do we have a microbiome? Well, obviously the microbes want us because we're awesome. We provide space, we provide nutrition, and we can even provide some protection. But why do we want them? Generally, they're doing something for us that we can't do for ourselves. A classic example is something like producing vitamin K. A lot of microbes can produce B vitamins, like folate, folic acid. There are a lot of scientists who are starting to understand that our normal microbiome actually can help train our immune system and keep it stimulated, not in an overstimulation way, but in a way that keeps it primed to fight an actual infection. We can also have our microbiome provide protection from pathogens. And so this is what we'll talk about when we talk about Clostridium difficile or C. diff. Patients often get C. diff infections because they've been taking antibiotics that wipe out the normal gut microbiome and allow C. diff to get in there and cause an infection. And like I've been saying already, your microbiome is influenced by your personal hygiene, by your diet, by the water you drink, by medicines you take, exposure to toxins, everything, right? Anything can change your microbiome. And when we talk about disruption of the microbiome, we call that dysbiosis. That's the word right there at the bottom of the screen, dysbiosis. It can be caused by numerous factors. Again, things like antibiotic therapy is the classic one, but even using antibacterial soaps, you can disrupt the microbiome of your skin if you have an injury, right? And that can allow microbes to spread in places they shouldn't be. People's microbiome changes as they change their diet. So they've done a lot of studies where people who generally eat an omnivorous diet transition to like a plant-based diet and their microbiome changes, and vice versa. Things like obesity can influence the microbiome. So lots of things can affect it. Now, when you change the microbiome, that's not always bad, especially if you have microbes that are still doing similar functions. So a change isn't always bad, but it can be. If you lose microbes that perform a necessary function, like produce a metabolite for you, if you have maybe a gap in your microbiome that allows a pathogen to colonize, that could be a problem. Or if you're no longer able to digest things that you want to eat. So they've done a lot of studies, again, in cattle, where if you disrupt the cow's stomach microbiome, they can't digest cellulose anymore. And again, scientists are really looking for these connections. There's a lot of correlation. So we know that dysbiosis is correlated with inflammation, with diabetes, with obesity. Does dysbiosis cause these conditions? Do these conditions cause dysbiosis? What is that relationship? And that's what scientists are examining. So those of you who find yourself interested in human medicine, but maybe don't want to practice medicine, like you don't want to be a doctor, this type of research program is really interesting. There are even people who are looking at the microbiome of cancer. Can you look at somebody's microbiome and predict whether or not they've got cancer? Because cancer can cause inflammation, which can change your microbiome. Can you predict? And so what we see on the left in this image is a cross-section cartoon of the gut. And on your left, it's healthy. You have all these microbes hanging out in your gut mucosa. You have antimicrobial peptides that it can be produced both by the gut microbes and your own body. You have IgA, which is your secretory antibody that protects your mucosal surfaces. And you have your immune cells that kind of surveil this area and try to make sure everything's going well. And for the most part, pathogens are kept out. Yeah, a couple can get in. Usually they're taken care of by the immune system, because if not too many get in, your immune system can clear everything up. But when you have a situation where you don't have a very robust microbiome, you have a depleted microbiome, you have far fewer organisms than you should, what they're trying to illustrate in this cartoon is you have a much greater chance of being colonized by pathogens. And the immune system may not be able to clean them all up. So it's really important to have a diverse and robust microbiome, not only in the gut, but everywhere you should. so that you can maintain a healthy host as much as possible. And then what they're trying to show here is how antibiotic therapy can change the microbiome. And I'm not going to ask you, like, remember all the details about this figure, what everything's changing. But the idea is these are patients A, B, C, and D, who were given antibiotics. And what we see, we'll just look at the top part. The bottom is very, very similar. So this is what the gut microbiome of the patient looked like before antibiotics, before. And really just what I want to highlight is that antibiotics, when you see the changes in the colors, that means the microbiome has changed. So in all of these patients, we see a change in the microbiome. In some patients, it's more significant. So patient B, right, had none of the green microorganisms really, except for like right there. And now there's a lot, right, after therapy. And after this patient finished their course of antibiotics, so before and after, their gut microbiome composition was completely different. Is that a bad thing? I don't know. Do I think it's probably a bad thing for this patient? Yeah, because some of the species that are enriched are things like E. coli and Klebsiella. Again, it's just something to be cognizant of if you're prescribing to a patient, or if you are the patient, that your gut microbiome could change, or it will change. But whether or not that's negative, I'm sure many of us have taken antibiotics and not experienced any gastrointestinal complications. Some of us may have taken antibiotics and had problems. It really just depends. And so you can see in some of the patients, patient A is the one that kind of recovered the best, I guess, though you still do have some enrichment of these E. coli and Klebsiella, but, your microbiome can change, and it will change when you prescribe antibiotics. And so that can be a major problem, or it could be not. So it really depends. And we think about the microbiome not only in what does it do for us, but how could we use it to help maybe treat conditions. So we can use, my favorite is treatment for Clostridium difficile infection. The scientific name for it is a fecal microbiome transplant. I call it repopulation. You take somebody's gut microbiome from their feces, and you give it to a patient who's got a Clostridium infection. And the idea is that if you have a healthy donor who is supplied a sample, and you give those microbes to your patient who has a C. diff infection, the healthy microbes should colonize the gut and help clear the infection. It actually works really, really well. There are a few different ways that the microbes can be delivered, through a suppository, through a nasogastric tube, or through a pill. I don't know which I would prefer. That has not been an issue I've had to encounter in my own personal life, so. But maybe we can use the microbiome as diagnostics. Alterations in the microbiome have been shown to be predictive of patients potentially developing something like necrotizing enterocolitis. And scientists have also identified that proliferation of certain species in the gut can lead to a predisposition for something like colorectal cancer. So perhaps, in the future, and maybe not in the far too distant future, maybe relatively soon, part of your screening processes as you get older and have to be screened for all of these things may include looking at your microbiome and seeing, do you have these microbes that could predispose you to cancer? Can we alter your microbiome in some way to hopefully reduce your risk? Be monitored, right? And I think some of us have also heard of and probiotics. Probiotics we probably more commonly heard of. These are supposed to be healthy microbes that can maybe balance the microbiome. It's been a few years, but Jamie Lee Curtis used to do these Activia like yogurt commercials. Like, yeah, okay, your yogurt's gonna make everything fine. Some people swear by probiotics. Sometimes if you do take antibiotics, a physician might recommend maybe upping your dose of yogurt or taking a probiotic. Most of them are gram-positive bacteria. Sometimes yeast can be included in them as well. People do use them to sometimes treat Clostridium difficile. That same idea of the fecal microbiome transplant. You should be, if you've got C. diff, right, and you put the good microbes in, perhaps you can kick out the C. diff. Maybe you could protect against salmonella infection or helicobacter. Therapy for autoimmune disease, you can use them in the mouth to prevent against cavities. Again, that's what they say. I've gone my whole life without taking probiotics and I'm fine. I know it's survivor bias, but that's okay. I had a student do a project on probiotics. This is one reason I'll never take them. He bought a number of different probiotic products and screened them for what organisms were in there. And most of them had way more organisms than it said on the bottle, and a lot of them were pathogens, and many of them were antibiotic resistant. So they're not FDA regulated, so there's no regulation. And we were laughing because we went to this one grocery store that I don't think is open anymore, but it was the only place I saw it. They had probiotic root beer, and they had probiotic popcorn. So I bought the root beer for my student. I was like, here, you have to try this. He goes, it's terrible. Well, yeah, sorry, but you have to try it because I won't do it. So you can get probiotic everything. We saw probiotic deodorant, popcorn, pills, kimchi, obviously all the dairy stuff. We lived right next to Whole Foods too, so there's probiotic everything. Prebiotics do not contain living microorganisms, but they're generally complex carbohydrates that are supposed to feed a healthy microbiome. So that's what prebiotics are. You kind of, feed your microbes, just like you would feed your pets. So perspectives to consider. Can disease be predicted by monitoring the microbiome? And these are not questions that necessarily have an answer. These are areas that are being explored by scientists interested in the human microbiome. What changes are most important? Can disease be treated or prevented by restoring a healthy microbiome? Will antibiotics be replaced if somebody has an infection? Can you just give them normal microbiomes and get rid of the infection? How do genetics, environment, and hygiene shape the microbiome? So again, those of you interested in human health but don't actually want to work with people, there are a lot of options for you to look at the human microbiome. Questions, comments, concerns about the microbiome? Okay, cool. If anything comes up, always just let me know. So this next section is about removing microorganisms. These are things that we want to do if we don't want microbes hanging around. And of course, a lot of these topics, especially in the first week or so, are going to be a review of BACT. So again, that's why I'll try to go through them a little bit more quickly. Antisepsis is using a chemical agent on living tissue. So an antiseptic is something you could put on your tissue. It does not remove all microorganisms. Disinfection is very similar to antisepsis. It does not remove all organisms, but a disinfectant would be something used on a non-living surface. So when you go into lab and you clean off your lab bench, that's disinfection. When you wash your hands, that could be antisepsis, or when put iodine before surgery or something like that. Oftentimes, it's the same chemical. You can have chemicals that can be used as antiseptics and disinfectants, but again, that key difference is an antiseptic is used on living tissue. Sterilization is totally different. Sterilization is the complete removal of all microbes, including bacterial spores. And here we typically do that through autoclaving. So anything that will not be like any solution or any substance that won't basically melt or catch on fire, we can sterilize through autoclaving. Things that might melt or catch on fire that can't be put through the autoclave, you can irradiate them. High levels of radiation can get rid of spores. You can filter sterilize liquids that might go bad. Autoclaving is preferred if possible. And you can always refer back to this table for these definitions. Here it just tells you some of the methods you can use to sterilize. Like I said, we typically autoclave, high heat under pressure, moist heat under pressure. But there are other ways you can sterilize as well. Okay. And again, if you've had a class with me, you know that I'm not like, please define ethylene oxide. Right? It's more of like the broad concepts. You can look up these details at any point. Disinfection, again, you can heat, you can use chemical compounds, hydrogen peroxide, alcohols, phenols can be used for disinfection. Remember, sterilization gets rid of everything dies, including spores. Disinfection, hopefully we reduce microbial load. We definitely are not getting rid of spores. Disinfection is done on something that is not a living tissue. And then antiseptics can be used on living tissue. You see alcohols are listed here again. People do use hydrogen peroxide on living tissue. Iodine, it's like betadine, things like that. Triclosan. Okay. Antiseptics can be used on living tissue. And then which ones work well against what. If you are disinfecting or you are doing antiseptic, you might want to look at this. For the most part, we can use phenol-based compounds to like clean lab benches, but 70% ethanol works pretty well. If you look at your standard hand sanitizer, I don't know where mine is. My kids took mine. They're obsessed with it. They can't get it open, but they love the bottle. It's like 65 to 75% alcohol. It's the same thing. So you can use antiseptics on yourself. You can use disinfectants on a surface. Generally, what's happening is with moist heat, so with the autoclave, the steam under pressure, you're denaturing proteins. So, remember back from chemistry, from any of your other biology classes, when a protein is denatured, it's pretty much done, right? You can't unboil an egg. You can cool that egg down all you want, but once you boiled it, it's boiled. Those proteins have changed and they don't change back. When you change the proteins in a living thing, you kill the living thing. If your proteins don't work, you don't live. And to make sure that our autoclaves are working, any facility that has an autoclave should be using a control spore-forming bacteria. So they'll test the autoclave periodically by putting spores in it and seeing if after the cycle the spores will germinate. And they should not. If they do, you need to fix your autoclave. So denaturing proteins is a very good way to kill things. For things that are heat sensitive, you can use various gases, you can use filtration. Ethylene oxide will basically form these reactive alkyl groups that prevents metabolism. But you have to do it for longer than autoclaving. You still would use the spore test as a control, but again, it just works by preventing the cell from being able to do its normal functions. Aldehydes, they will also denature proteins through reactive groups. At low concentrations, you can use aldehydes to be bacteriostatic, prevent bacterial growth. They're toxic to humans. as you might expect. An autoclave is probably also very dangerous for a person. You do not autoclave people. So people do not get sterilized. Not like in, you can if you want to be, but that's totally different. That's a reproductive sterilization. You actually don't like remove all the microbes from you, because whatever you do to remove all the microbes from you will probably kill you too. Oxidizing agents will form free radicals, which can kill microbes. Again, you have to be really careful with this. Oxidizing agents will damage your cells too. So free radicals will damage DNA and proteins. Halogens will also cause protein issues. They will oxidize essential enzymes, but they will do this to you as well. Phenol compounds disrupt lipid membranes. You have a lipid membrane. So there's a theme here, right? Be careful. You could hurt yourself. And ammonia compounds will denature cell membranes. At lower concentrations, you can use them to stop bacterial growth rather than to kill microorganisms. Alcohols also work by denaturing proteins and disrupting lipid membranes. That's why maybe you found out during the pandemic that if you use a lot of hand sanitizer, your hands get real dry and cracked because you're destroying your own cells. So feel free to use hand sanitizer. Like, I use it, no problem. Not all the time. You can use soap and water too, but you can use hand sanitizer in a pinch, but it works mostly by disrupting the lipid membrane. As I told you, some of the chapters are super short. And these are things that we'll cover again in the lab because, it's the lab. And we have to make sure that we do these things. But so let's get into some actual medical microbiology. How do we diagnose infections? As we talk about each class of organism, so we're going to talk about bacteria, then viruses, then fungi, then parasites. We will talk more about these concepts, these principles of diagnosis, because we're going to have to talk about how you diagnose every type of infection. But broadly, there are tools that scientists use to diagnose infection. Those of you who are going to be working on the patient side, you may not be doing as much of this diagnostics. So why do I teach it to you? Well, one, because not everybody in the class wants to work on the patient side. But two, as a physician, you are responsible for ordering tests. And as we start talking about the different groups of organisms, we'll play a game where you will be physicians that have to order tests, but you have a budget. You can't just order whatever test you want. So you have to know the right tests and what the tests will tell you as a physician, even if you're not the one actually running the tests. So a common way that we can at least diagnose that someone has an infection, and depending on the type of organism, we can start to maybe get an idea of what the infectious agent is using microscopy. There are a number of different types of microscopy. I know you covered this in Bac-T because I taught Bac-T over the summer, so I know you cover it. Most commonly, we're using bright field microscopy or light microscopy, where you have a illuminated background and your sample is darker than the background. For lots of things like bacteria, you can't actually see them unless you stain them. So we have all the different types of staining techniques. And those of you who took Bac-T lab with me, you're going to hate this. We're doing gram staining a lot. Okay, you're going to do a lot of gram staining, so review. So it relies on visible light. Light microscopes are relatively inexpensive. Most labs will have one. But they do have the lowest resolution. You can't see super, super small things. And that's just because of the physics of light. Dark field is similar. But you have an illuminated sample and a dark background. Sometimes that can be helpful if you have really thin organisms you're looking at, like spirochetes, or if you want to maybe get an idea that perhaps the cell has a capsule or something like that. It is a little bit increased resolution, so you can see smaller or narrower things. So spirochetes are really long, but they're super, super thin. And so using dark field microscopy can allow you to see them better. We also have phase contrast microscopy, which can allow the imaging of internal structures, particularly if we're talking about fungi or protozoans. Another benefit with phase contrast is, that's how phase contrast works. It doesn't kill the organism. The light is not as concentrated as with bright field. And so phase contrast can allow you to visualize living structures as well. And again, I'm not going to ask you all the details of how do all the microscopes work. Okay, you can look that up. You can also do fluorescent microscopy. It's a variation of light microscopy. You use fluorophores or fluorescent compounds to tag. You can tag just cells in general. If you have eukaryotes, you can tag the nucleus with one tag and other parts of the cell with another. And you excite the fluorophores using one wavelength of light, and then you can read the emission on another. And so this is actually microscopy that I did as a grad student. It doesn't show up super well on the screen, and I'm not going to turn off the lights because I won't figure out how to turn them back on. But when you look at it on your own screen, there's like red and green. So this is a type of staining where green cells are living and red cells are dead. So the living cells are able to exclude the red compound, but the dead cells aren't, so they turn red. So you can do fluorescent microscopy. Not a lot of labs have this, so I don't imagine many of you working in a clinical setting will have access to electron microscopy. But if you do go into research, or if you end up working for the government is always hiring microbiologists. I did my postdoc with the Army. They have like more money than they know what to do with, so they have things like this. Electron microscopy doesn't use visible light, it uses beams of electrons. Very high resolution. With certain kinds of electron microscopy, you can actually see viruses. You can see viral particles with electron microscopy. So this is an electron micrograph. I didn't do the electron microscopy because they paid somebody to do it for us, but this was a biofilm of Staph aureus that I grew, visualized under scanning electron microscopy. So again, unless you work for a big fancy lab, you probably won't have access to this, but this is the only way that you can actually directly see a virus. With light microscopy, you can see bacteria. You can certainly see fungi and other parasites like the protozoans, and of course the helminths. Sometimes you can see with your naked eye because tapeworms can be like 20, 30 feet long. You can see that. For viral infection, one of the challenges will be, those of you working in clinical lab settings, you'll often look at host cells. So you'll look at cells from the patient and look for changes to that cell caused by the virus. So you won't see the virus itself, but you'll see the effects of the virus on the host. One of the classic examples is in a pap smear, which looks for changes in cervical cells that could indicate the development of cervical cancer. And the reason I bring that up in a microbiology class is because 99.7% of cervical cancers are caused by a viral infection, by HPV. So you're really looking at viral changes to the cell. So it's very, very, very rare that a patient will develop cervical cancer without HPV infection. So again, as the clinician, right, as the clinical laboratory professional, you are not going to see the virus, but you'll see the virus effects. And so again, larger organisms are easier to see. And I hope that you never ever happen to be the physician diagnosing the tapeworm infection. But like there was a guy that turned up in Fresno a few years ago that had a tapeworm he pulled out of himself and showed up at the hospital like, what is this? So sometimes you may, but smaller organisms, most infections, right? If you want to visualize the infectious agent, you will have to use microscopy. So on Wednesday, we will pick up with more about examining and how we would do culture and things like that. And we'll also have our first lab session on Wednesday. And so please make sure you look at the lab PowerPoint. It's going to be very much an introductory lab. Next week, we'll start doing actual lab techniques, and that will continue throughout the rest of the term. There is 1 week in October where we won't have lab, October the 4th, the first week of October. We also won't have lecture on Wednesday and Friday. I will not be here, so I will record some lecture videos for you. But otherwise, I'm going to do my best to record class every day. I'll post the videos so you can watch that and refresh yourself or use it as a study tool. And I look forward to seeing you all on Wednesday. So if you could bring your names back up, I'll give them back to you until I learn your names. It won't take me that long, but I'll get there.